Hey guys, welcome to this VR video about bioproduction. In this video we're going to go through the steps that will lead us from the mouse to a biomanufacturing of a protein, which is going to be a monoclonal antibody. I choose this example because it is very relevant regarding the market of biopharmaceuticals, and that monoclonal antibodies are now proteins that are widely used in human therapeutics. So first, why the mouse? What you want is to have a human-like monoclonal antibody directed against an antigen. This antigen, which is the target of the antibody, is what makes you ill. In the disease, it is something that makes you ill. It has a pathophysiological role. It is a protein that can be expressed or can sometimes not be expressed normally inside the body, but in this context, it makes you ill. So what happens if you actually inject an antigen, this antigen, to a human in order to get a monoclonal antibody directed against it? Aside from ethics, bioethics, it's just that there's few reasons for anything to actually happen. The antigen being human, when you inject it inside a human body, it's very likely that nothing will happen at all, since it is recognized as a self-antigen, what you call a self-antigen. The immune system recognizes it as part of the human being and will not react against it. On the other hand, if you inject it to a mouse, then you will get an antibody response, an immune reaction. So you have your mouse. Your mouse has billions of B lymphocytes. B lymphocytes are the cells which are responsible for the synthesis of antibodies. What you want is to select the few antibodies that actually relate to the antigen that you've injected. But you can't choose. What happens is, once you get your cells inside the syringe, you actually don't have an access to what these cells are directed against. So you have to find a way to split these cells to be able to choose which one is the best for your treatment. There's a rather simple technique which is very old but works very well, is to dilute them a great deal. What you do basically is that you have a well plate like this one, in which you have wells. These wells are filled with diluent solutions, and your cells are being diluted and split between the wells. So basically, if you dilute them enough, you have most wells with actually nothing at all inside them, nothing but diluent. In some wells, you'll actually get one cell in a few wells, and in others, very rare, you can have more than one cell. These ones aren't interesting to us today. Also, what you have in the well plate is a system that reveals the presence of interesting antibodies. These antibodies are basically the ones that react against the antigen. So if you put a system, for example, a system that gives you fluorescence, a fluorescent signal, when you have an interaction between the antibody and the antigen, you can tell in which wells you actually have the antibody that is capable of binding to the antigen. So now I know where to find my antigen reactive cells. There's a few of them. Here I drew 1, 2, 3 cells. I have 3 candidates for my therapeutic purpose. What I need to do is to pull out the information. So I need to expand these cells, take out the genes that encode my protein, and then we'll see what to do with them. So first of all is, how do I expand my cells? I use flasks such as this one with a culture medium, which allows me to expand these cells, which have their own genome. What I want is to get only the gene encoding my protein. So to do that, I'm going to use a molecular technique which is called polymerase chain reaction, very famous, which has evolved a great deal the last few years, which is an iteration of these 3 steps. First of all, the medium is going to be heated. You have your DNA here, you have 1 and 2 genes, and what you want to do is to separate the strands, the DNA strands. Let's put it this way so we can see nicely. There. So the strands, you have 1, 2, 3, 4 strands, which are held together by hydrogen bonds. When you heat the tube, you separate the strands. So you get 1, 2, 3, 4. These strands are now open for the polymerase reaction to happen. You have DNA polymerases here as these little dots. They're going to use the nucleotides inside the reaction medium to fill in the blanks, to complete the strands that have been left open. Then you cool down the medium. The temperature goes down and DNA strands actually fuse together according to base complementarity. This is the 3rd step. And at that stage, you have doubled your genetic material. So what you want to do is to go all the way through the steps again, do another round in order to increase the genetic material. This genetic material you're going to be needing to understand what your protein actually is, to understand what it's made of. DNA encodes proteins. 3 nucleotides make a codon, and a codon corresponds to 1 amino acid. So this is the reason why taking out the information out of the genes will lead you to know the structure of your protein and allow you to modify it. You want to modify the protein because it's actually a murine protein, and you want to turn it into a human protein. So you need to have an access to the sequence, protein sequence, in order to change the amino acids that resemble those that are used by mice in certain structures. Sounds a bit complicated, but in fact, we have 20 amino acids, about 20 amino acids that we use in our proteins, which are the same basically as those used by the mice. But they use them in a different order. Sometimes they have sequences that they use which are recognized as foreign by human immune system. So you want to switch some of these residues, some of the amino acids, you want to change them in order to make it a protein that resembles very much an antibody from a human. This avoids some of the problems that you can encounter when administrating a foreign protein to a human body. So now we have the genes, the 2 genes that encode, one the light chain and the other the heavy chain of the monoclonal antibody, but we still don't know what's inside them. We're going to be needing a sequencer, the SEC3000, which doesn't exist as far as I know. This machine here gives you the information concerning the nucleotides inside the genes. So you have a graph here with 4 colors. There are 4 different nucleotides, A, T, G, and C. So let's say that maybe A is green, G is black, and so on. This machine here cuts one nucleotide after the other and gives you peaks. Why are there several heights of peaks? Well, you have, in general, a single height, which is the unit. It says 1 times A, 1 times G, 1 times C, etc. When you have twice the same nucleotide in a row, then it gives you a peak which is double the height. This is the reason why some peaks are higher than the others. So this reads For example, if we say that A is green, it goes like A, then G, then C, and so on. Oh, I just realized that there was a mistake because these two peaks here should be like one big red peak. Thank you for noticing. Once you have the sequence, you know what this is going to transform into, what is going to be translated into. So you know the structure of your protein. Then you have an access to databases which help you choose which amino acids to change in order to have a human-like protein. So basically, from now on, you'll be needing a computer, and a powerful one, because you're going to have to build the gene sequence that you're actually going to use to produce the antibody. So you design either a plasmid or a linear sequence, depending on whether you will transfect stably or instably your cell. And once this is done, you have it fabricated for you, and then you have to knock it inside the cell which is going to be used for the production. Now this cell has nothing to do with the B lymphocyte from the mouse. It is a cell which can be from any organism, ranging from a plant to human cell. And this cell is going to be your manufacturing facility. Your genes have been knocked in. So you have the sequences encoding your protein inside the cell, and this cell is actually able to synthesize the protein that you need here. So what you need to do now is to put it inside a bioreactor, this thing here. This is where you'll grow your cells, and that will allow you to have large amounts of antibodies, which are going to be used for the tests. In vitro testing, then in vivo testing, preclinical ones, which basically means testing on animals, like the mouse you've used at the beginning of the experiment. And after that, the clinical batches, which are going to be used for the clinical trials. Now, if we go inside the bioreactor to see what it's like in there, I'm going to take you inside the bioreactor with me. There. Bioreactor is the place where you grow your cells. Inside it, there's mechanical agitation, stirring, which allows for the nutrients and gases to mix inside the reaction medium between your cells to nourish them, allowing them to produce the antibody. There, for example. These antibodies, once you have a high concentration, or at least once you get the maximum concentration without having too much heterogeneity, then you have to empty the tank, or at least take out part of the reaction medium, and then you'll have purification steps to avoid any residual cell material. We call them host cell proteins. Purify the antibodies in order to be able to formulate them inside vials, which are to be controlled and then released to carry on for the experiments or the clinical trials. That's the end of this first video. Thank you for watching. I hope you enjoyed it as much as I did, and see you next time.